Hello, so I am back to talk about lysogenic conversion. I'm going to talk about the differences between lytic and lysogenic replication, as well as the role that lysogenic conversion plays in the development of pathogenicity in bacteria. So first, lysogenic conversion is a process that occurs between a bacterium and a phage that is also often beneficial for the bacteria. In lysogenic conversion, the phage inserts specific characteristics into the bacterial genes, causing the bacteria to have a better survival rate. The ability of some phages to survive in a bacterium as a result of the integration of their DNA into the host chromosome. The integrated DNA is termed a prophage. A regulator gene produces a repressor protein that suppresses the lytic activity of the phage. But various environmental factors, such as ultraviolet radiation, may prevent synthesis of the respirator, which leads to normal phage development and lysis of the bacterium. The best example of this would be bacteriophage lambda. So, three differences between lytic and lysogenic replication of bacteriophages. So, the first one, in lysogenic cells, the spread of viral DNA occurs through a usual prokaryotic reproduction, where a lytic cycle is more intermediate in that it results in many copies of the virus being created very quickly and the cell is destroyed. For lytic, for the second one, lytic, the DNA of the virus doesn't integrate into the host DNA, where lysogenic, the DNA of the virus integrates into the host DNA. And then the third difference, lytic is the cellular mechanism of the host cell is totally undertaken by the viral genome, and lysogenic is the cellular mechanism of the host cell is somewhat disturbed by the viral genome. And lastly, what role lysogenic conversion plays in the development of pathogenicity in bacteria. So when a lysogenic conversion occurs, it can change the property of the outer membrane of bacteria. Thus, it is possible that other phages can infect the bacteria more easily. Sometimes lysogeny increases the pathogenic capacity of infected bacteria. When the virus infects the bacteria, it can also release some toxins, like exotoxins, into the bacteria which do not have it. These exotoxins or virulence factors can spread by the bacteria when it infects others and can cause disease. Lysogenic conversion can also make changes in the colonization property of bacteria or resistance to the antibiotic. Two examples where lysogenic conversion has contributed to pathogenicity. So during lysogeny, the virus integrates its DNA into the genome of bacteria. In this way, the genetic material of DNA, which is called prophage, will be carried toward the subsequent daughter cell. It happens when a temperate phage induces a change in the phenotype of the infected bacteria that does not belong to the phage cycle. Some phage carry genes that encode toxins. They integrated into the bacterial chromosome and can cause the harmless bacteria to release potent toxins that can cause disease.